All right. Thanks, everyone. As Jim said, my name is Gene Civillico. I'm a program manager in the Office of Strategic Coordination, which manages the Common Fund. I specialize in neuroscience and bioengineering and data management, and among other things, I run the SPARC program. And it's been my privilege to talk to this group about SPARC twice before, actually. In September of 2019, I spoke to you about a couple of new initiatives that we were adding to our program. And a year before that, in September of 2018, you may remember at the Cloisters when we brought in Terry Powley from Purdue University to talk about his work untangling vagal signals from the stomach for better vagal nerve stimulation. So my purpose today, and Denise, if you could go forward one. Our purpose today is to talk to you about a concept clearance for a set of new initiatives that would comprise a stage 2 of this program. And they're named and very briefly outlined here on this slide: SparkV, SparkO, and SparkX. And before we can talk about why we want to do these initiatives and how they follow on from what we've done so far and how we think they could amplify the impact of the program before it ends, I just wanted to refresh your memory on what the program is about. In some ways, the previous presentation is a good setup. You know, you could think of us as eliminating the stimulatable nervome, if you will. It's the sort of device equivalent of IDG, obviously with a lot of changes because devices are not drugs. If you, Denise, if you could go forward twice. One more. Thank you. Yeah, so the SPARC is a program, in short, about neuromodulation below the neck. It's about, rational mechanistic targeting of device interventions, about bringing the excitement that's happening in neurotechnology with things like the Brain Initiative and making sure that it gets applied to the myriad of problems that people have that are not located in the brain. Cardiac problems, gastric problems, bladder problems, inflammation problems, et cetera. And with SPARC over the past 5 years, we've gone out and grabbed communities that have not engaged with each other enough, the bioengineering and basic research and clinical practice, and have pulled them toward each other in the name of kind of next generation device approaches. The initial challenge that animated this program is summarized on this slide. Companies are making these devices, and they're working sometimes, and they're not working other times. And there is a lot of mystery associated with the efficacy of these devices, and the mystery is not surprising because the science base underlying them is incredibly primitive and simplistic. So there was an urgent need for kind of clear mechanistic targets with a rational basis in anatomy and physiology. And your slide 13, which is in your background slides, and I don't want us to advance to slide 13, but slide 13, if you get a chance to look at it, shows a really good quantification of how understanding more about a therapy leads to approval of the therapy and acceptance of the therapy. So we decided, that NIH was going to try to help with this. Denise, if you go back now to the previous slide, I apologize for the out of order slides, but it's going to go away pretty soon. So the program architecture, once this basic need of let's advance next generation bioelectronic medicines, the program architecture was informed by a workshop from 2013 and then a research roadmap paper that followed from it in 2014. And these activities I elaborated sort of 4 critical priority areas to advance this field. And at the time, they were calling it electroceuticals. We don't call it that anymore. We call it bioelectronic medicine now. And the 4 critical areas are shown at the right of the slide. So number 1, biological maps of structure and function. Where are the neurons? What are they doing? The same question that we are asking with all of our brain atlases needs to be asked about all the other neurons that are not in the brain that are also impacting function. Advancement of interface technology. How do we interrogate and manipulate these neurons? early establishment of therapeutic feasibility. How can we test quickly whether we have a mechanism that can make a difference for a disease or condition? And finally, collaborative data mining and standards. How can we put our research output out into the world in a more reusable and modular way so that we can learn new things more quickly? And we envisioned that all of this would be enabled by an emerging community of bioelectricians, which is just another way of saying a particular type of bioengineering or biomedical researcher. So to get to these three goals, the program was designed in sort of common fund style with 4 interdependent thrusts. So Denise, now we're going to go forward 3, if you wouldn't mind. Yes, perfect. And the thrusts are here in the 4 different colors, and they loosely map to the 4 priority areas from the previous slide. So mapping, data collection across multiple organ systems about innervation, development of new sensing and stimulating technologies, data-rich clinical studies, partnerships to drive studies in humans, and an online data and resource center with core functionalities of data management, map or knowledge synthesis, and computation and simulation. We also added a 5th initiative in 2019 in association with the HEAL program, supporting additional anatomical and functional studies of neurovisceral circuits carrying pain signals. And in your background slides in slide 15, there is a snapshot of the timeline of these initiatives and how we added them on, integrated them with each other, and what their approximate sizes were, if you want to have a look at that at some point. So we stood up the program. It's a common fund program. It's a collaboration between NINDS, NIBIB, NCATS, and NIDDK. And it fits very well in the common fund because of the way it cuts across IC missions. You know, it cuts across therapeutic areas. It cuts across technology development and basic research. It's neuroscience. It's cardiac. It's digestive. It's bioengineering. You know, it's all of those things, and so it's a great place for us to contribute in pulling areas together. Now, in the time we have today, can you click forward one, Denise? It's not possible to go through everything that the program has done. One more time. Thank you. But this is just a smattering of some of the papers that have come out recently or not so recently, supported by our teams. There's a lot more in the background slides. I think particularly in the background slides, we've pulled out and blown up some figures and some title pages from some papers to give you an idea of the really cool things that have come out of the program. I also encourage you to check out, and you can do it right now if you want to, SPARC.science. That is our data portal where you can see not only raw data sets that have been curated and standardized and made available for reuse by our investigators, but also links to our preprint channel for the program on bioRxiv, our shared protocols channel on protocols.io, as well as links to a PubMed page that collects all the traditionally published work that we know about based on a search of award numbers. So I encourage you to take a few minutes and look at those things anytime you want. The SPARC.science is really, I think, a vision of the future in terms of how we're going to display and share and talk about the products of research increasingly going forward. Denise, could you go back to? Thank you. So in these past 5 years, we've moved the ball substantially on generating organ innervation maps, new tools, and new clinical studies. But the original rationale for the program was to advance therapeutics and really to try to do that in a transformative way, not sort of someday, you know, far in the future, but soon on the time horizon of a few years. And so we've been asking ourselves, you know, as we come to the end of our original, you know, budget envelope, which is this year, is there something more we can do that would be more transformative? And we've been doing a lot of listening this year. We've undergone a very open consultation and input gathering process, all virtually during the past several months. We posted an RFI in the summer inviting people to tell us about ideas for ambitious collaborative goals in bioelectronic medicine. We had an ideas summit earlier this past fall, where we gathered together the 3 major neuromodulation companies, researchers and senior leaders from those companies, as well as a set of smaller companies and some translationally minded academics. And we discussed, the roadblocks to advancing the field and different models of public and private funding working together. And all of this input produced, I would say, 2 main principles. And so beginning next year, and what we're here to talk to you about today is, pivoting the program in the direction of those principles with 3 concept initiatives that I'm going to tell you about today. So I'm going to tell you about 2 principles that we have distilled out, and then 3 initiatives that are going to spring from those principles. And we're just going to leave this slide up while I talk about the principles. So the first principle is focus. And actually, Denise, could you hit the right arrow just one time to pop those images up? That's great. Yeah. And so what you can see down at the bottom of this slide is really both the success and the problems, right? So SPARC-funded teams have learned an incredible amount about these little brains that are all over the body. But when you look at these images, it may or may not be obvious to you guys that the left one is a heart, and the second one from the left is a pulmonary, and the middle one is a stomach, and the next one is an intestine, and the next one is a ganglia associated with the bladder. So they're literally all over the place. They're all over the place in the body, hearts and stomachs and adipose tissue and rats and mice and pigs and cats and humans. And we've developed an entire ecosystem around curation and sharing this data. But the power of what we've developed is getting diluted because it's spread across so many different application areas. And so what we've realized is that to really show the value, we need to focus. And that's what SparkV is about. And I'll get to that in a minute. So first principle is focus. The second principle is to be a bit more transformative in the way we think about getting really path-breaking device innovations, to patients. We've worked with industry a lot in SPARC. We have three whole slides about it at the end of the background. Easily 10 or 20 active industry partnerships between people that we've funded and companies in this space. And this partnership model is used by Brain and other programs. It's fairly well established at this point. Essentially, we put up a list online of companies and devices and contact information, and it's basically a menu that an academic investigator can pick from. Then they have contact of who to reach out to. They have template disclosure agreements and other types of documents that they can fill out together with the company, and then they come in and apply for NIH funding in the semi-normal way with a letter of support from the company, and the money goes from NIH to the academic researcher, and the company provides sort of in-kind support in terms of either engineering time or device time. The main problem with this, and it works pretty well and companies like it, and researchers are, they find ways to work with it. The main problem is that it is mostly larger companies that are interested in this, and the possibilities get filtered through their strategic goals. So oftentimes they're pursuing kind of incremental improvements to their existing products. Even if they have a really interesting strategy, that strategy can change in the middle of the grant period. And then suddenly that in-kind support isn't there anymore, and the investigator is on the hook to NIH to get certain things done, but their partner, is less interested than they were. Often it is smaller companies that larger companies are looking to prove out the risks of new strategies. So you might say, well, then obviously you should be working with smaller companies. But the problem with that is that by the time a small company exists, they usually have a pretty clear idea of what they need to be doing. So you don't spin out a startup and say, now what can we do with our startup? You spun out a startup because you had a thing that you needed to do. And then once you get that startup going, you need to do that thing because you have investors that are waiting for you to do that thing. So existing smaller companies need to pursue their founding goals. So what we are thinking is that we should be looking to lay the groundwork for the creation of new small companies by providing academic researchers with a neuromodulation platform that is better than their homegrown research-grade devices and not tailored to the legacy strategic objectives of larger companies. So we want to focus more on the academic small company hybrid ecosystem, which is where we think that next generation strategies are likely to emerge from. So both SparkO and SparkX are intended to target that need. So I'm going to take now each of the V, O, and X and expand each of them just a bit as concepts. And Denise, this is going to be our last big jump. If you could go forward 3 to slide 7, and then we're going to stay in order after that. Great. So one more back. There you go. Thank you. So what we want to do with SparkV is take the power of this ecosystem that we have up and humming now and apply it to one area of focus. And we believe this focus should be the human vagus nerve. The sub-initiatives are described here, and we would use our existing data infrastructure to host this. The goal of SparkV would be to create circuit-level descriptions of the anatomy and physiology of the human vagus nerve at the sort of subway level, which is what that image is meant to evoke there. Not at the electron microscopic level necessarily, but at the level where you would engage them with devices. So we'd like to have a standardized sample to image to data pipeline and digitize the interface points of, ideally hundreds of human vagal nerves, left and right, male and female, as much cohort diversity as we can figure out. That would be one initiative. The second initiative would be to take advantage of the large population of people that are already walking around with implanted vagus nerve stimulators that have never been set to anything other than their initial settings, and actually get people to engage with their clinicians and go through a standardized set of parameter sweeps within the safety protocols that are allowed by the FDA, and actually systematically explore the effects on heart rate and gastric motility and mood and anything else that people can make the case for, and actually put out there a sort of functional map of what you can actually do when you perturb the vagus nerve in humans. I think this is totally possible. And the third sub-initiative would be to continue the development of our portal to host the results of this work. So the portal would go forward with this kind of a dual mandate to serve the SparkV initiative, and then also to be able to stand alone within 3 years as a specialized repository for neurostimulation relevant data, especially in the periphery. The new data management policy from NIH, as many of you might be aware, recommends that data from NIH-supported work be deposited in a specialized repository if there is one. And Brain is funding a bunch of these, but they're mostly for brain. And there is really no specialized repository for the kind of data that we have been and expect to continue to be collecting. So we think there's a good reason for a final push to make this a sustainable, independent resource. And if you take a look at it, it's really not far from being that. So in case you're wondering why the vagus, Denise, next slide, please. These are the reasons. VNS is sort of in the sweet spot. It is an established treatment modality, but we still have so much to learn about it. It cuts across the mission areas of all of our partner ICs and several others. It's been the site of a lot of our most exciting and most on-mission stuff over the last few years, and there are particularly exciting opportunities in neuroimmune and cardiac hypertensive applications, both of which people are poking around those areas. with regard to vagal nerve stimulation, but they really, it seems unlikely that they will be successful without a leap forward in terms of understanding. So that's SparkV. Now I want to return to the theme of pushing innovation and moving new kinds of devices to patients, and just quickly tell you about O and X, and then we'll be able to talk for a bit. So next slide, please. As I mentioned before, one of the real needs that our neuromodulation-focused researchers have told us about is the need for more flexible clinical-grade platforms that are strategically independent from existing major players. We've heard articulated that academic researchers need a clinical-grade device platform that is available to anyone, but that there's no private incentive for anyone to make that. Many of the characteristics of such a platform are common to all neuromodulation applications. So this is an ideal place where we could help, and we can do this as a collaboration with other neurotech-flavored programs, such as BRAIN, but not just BRAIN. And if we do that with the folks that we have that are interested in doing that, it will help those projects to transition to other funding in a phased way, which is something that we always worry about. So if we co-design this sort of exit strategy with other programs, then they will be able to take it over because we can make sure that it fits our objectives and their objectives. And the basic concept is the second bullet here, that applicants will propose to create interoperability specifications and compatible modules for stimulation, recording, waterproofing, interfacing with leads, coatings, batteries, and these things can be combined into custom hardware profiles, and then academic researchers can use the FDA's right of reference system to build on what other people have done and not need to come in from scratch every single time with a new device. We can link the modules into a consortium to support interoperability and a shared roadmap for the platform. So that's SparkO. We have a lot of excitement around that among the NIH program folks, as well as in the outside community. And finally, SparkX, calling back to my comments before on industry, one of the insights that Denise, could you go forward one? One of the insights that came from the Ideas Summit that we had in the fall, and I didn't mention this before, is that a challenge-based incentive from the government, some of the industry folks felt that would give them an incentive to reach out to their counterparts in other companies to work together on something without requiring their companies commit to the overhead that comes with entering a government funding arrangement. So they could just start working and try to do something really amazing, and then if they won this challenge, that would bring a lot of cachet, and people would be excited about it. So we want to explore the possibilities of doing this. We called it SparkX, referencing the X Prize competitions that you might be familiar with. The idea would be a series of increasingly difficult prize challenges that would build up to demonstration of some completely new capability that we can imagine now, but just hasn't, we're not seeing it coming. One good example is what I have here as a challenge goal, demonstrate independent vagal modulation of multiple endpoints in a large animal model. And these prizes would reward any U.S. entity, for profit or otherwise, independent or affiliated, who's able to hit certain benchmarks that are defined in the challenge. And there's a lot of flexibility and possibility with how these things are being used, and we have access through an NIH MOU to a center that NASA has set up where people assist program staff in defining effective challenge goals, and so we will engage everyone that we can to figure out how to do this. The staging strategy would allow us to hold back on major commitments until we're confident that that it's a good idea. So, for example, the later stages with the big payouts could be contingent on us getting a winning entry from the planning stage, which would have a smaller payout, so that the sort of larger payouts are gated by success in the smaller payouts. And that is SparkX. So if you, I think one more time, Denise, if you could click. So I told you about our plans to launch SparkV, SparkO, and SparkX as a sort of coda on the SPARC program, and to continue the development of the SPARC portal to support them. We think that this would really put an exclamation point on this program and make sure that these ideas are going to animate this field, through fiscal 24 and well beyond. And I'm happy to discuss.
Okay, thank you, Gene. We're running a little short on time, so we're going to just dive in with comments from Dr. Kenny, then Dr. Chang.
Yeah, I'll make my comments pretty short. I'm a huge fan of the SPARC program. I wasn't aware of it until I rotated onto this council, and I believe maybe it was even one of the first council meetings that the SPARC program was presented. From my perspective, I think it's incredibly important. The potential is also incredible. The peripheral sensory neurons that are delivering information into the brain are likely involved in a whole host of diseases. We know far too little about how ascending information gets into the brain and descending autonomic information, parasympathetic transmission, gets out. Understanding the sensory input is likely to be relevant to a whole host of diseases. So these three components, I think, are the logical next steps. I am a real fan of SparkV to really just try and understand the basic mechanisms of sensory neuron information delivery into the brain. Obviously, the other two components are outstanding. But, I think that the potential payoff here could be beyond just neuromodulation using devices, which obviously this is what the program is targeted toward. There's a whole host of receptors and genes and proteins expressed in those ganglia outside the brain that are accessible. You don't need to get into the brain. And likely small molecule drugs that are already in human are acting in those ganglia. And we have no idea what they're doing. And so the more information I think we have about the vagus and the other sensory ganglia in the body, I think the more likely we are to essentially stumble upon mechanisms through which existing drugs work and mechanisms that we can take advantage of for new drugs. So like I said, I'm a huge proponent. I think this is a great initiative and I'm fully supportive.
Thanks, Paul. Linda?
I concur with Paul, and I also am very big fan of this program. We both reviewed it previously, and I think the new initiative, the VOX, are really nice addition to what you already have established. I just have some small questions about each one of this. So, as a clinical neurologist, I've been using vagal nerve stimulation, and I know how effective it can be in treating certain conditions like epilepsy, for example, and I know it's been used for treating depression, anxiety, and all that. And below the neck, it goes to so many different organs, and so the vagus nerve is primarily parasympathetic. Do you have plans to also develop something similar for the sympathetic nervous system in the periphery? So that's for the first part. And my second question is about the open science. Actually, it's not kind of a question, but I think it's really great that you bring together the open science and data sharing approach so that everyone can benefit from that. But it was kind of unclear how you could bring this to bring in more bioengineers from the field to further, grow this initiative, or is it just for people that are already funded to kind of collate their data? And then my third question, the last one, is about the X Prize. It's very interesting and fun idea. Were you planning to have more than one winner, or just because it seems like a big pot of money? So, or is it just one prize? Those are my questions.
Gene, do you want to go ahead?
Okay, I can take those in turn, Jim, if that's okay.
Sure, yes.
Yeah, it's a great presentation. What you did is wonderful. Yeah.
Thank you. So we have, SPARC-funded people have studied many sympathetic pathways over the last 5 years, and there's a lot of cool stuff that's been done. When we're focusing on the vagus, we're really doing it for essentially practical reasons because it's an established route of delivery of this therapeutic modality, but it still has upside in terms of what we could learn about it to make it better. So it's sort of a practical judgment that if we demonstrate value for VNS, then other people will follow in our footsteps in other, sympathetic pathways. And we certainly recognize that, I think someone on our working group actually brought this up that, well, suppose we get really good at delivering parasympathetic stimulation, then everybody's going to be, then we'll have cures for anything that requires parasympathetic, activity, but then we'll have the opposite problem. We won't be able to control the sympathetics. And, if we get to that point, that would be a great problem to have, because it will be very easy to argue that we need a, SparkP or SparkS or whatever. It won't be us, but it could be someone else. So that's kind of how we look at that. To your second question, the data sharing, we have our hands full with curating and publishing the data sets from the people that we've funded, but we do have a sort of queue of folks who are excited about this and would like to put their data up on spark.science. And one of the things that we would like to do, in the final 3 years of the program is make sure that our portal people are ready to do that, that they're resourced to do that they've wrung the inefficiencies out of the data pipeline so that it actually is possible for them to take care of as many people as we hope would like the services. The last question about the X Prize, I don't think it needs to be. We haven't decided, and I think it can be described in the challenge solicitation. How many winners there will be, and how that would be handled. I can see advantages to doing it either way.
Yeah, Linda, I'll add to that last one. The division is responsible for challenges, for prizes, for overseeing the policy processes, approval. We work with general counsel on every proposal that comes through. So we would help folks design whatever it is. We have multi-stages, multiple winners. It's really a fun way to design getting something done. So we have time for just a couple of more comments or questions from council before we take a vote on this. If folks can please put a comment, put your name in the box.
Dr. Van Gelder, you are unmuted. Please
Good.
Go ahead.
Okay.
Great. Thank you, Gene. That was a great presentation and really exciting science. I have two questions, I guess, / comments. One is, have you thought about the name of the project right now relative to where it's gone? It's kind of turned into an autonomic stimulation or even parasympathetic autonomic stimulation project. And as you know, there are other major areas of nerve stimulation work. Probably the most successful is cochlear implants. I have some experience with retinal implants and Argus 2, which is one of the few FDA-approved devices. Obviously TENS units and peripheral pain stimulators. It seems like, pardon the pun, but I would hate to see what happens in Vegas stay in Vegas, and is there any way that we can kind of broaden this out to create a broader community, or change the name and say that this is an autonomic stimulation project? And then the second question is really around the X Prize versus the other ways of achieving the same ends. And I think it's very difficult to know what the small business community is because no one actually organizes this and says, what is the small business landscape for this particular type of technology? I've been surprised many times with how much activity there already is in areas that I would have thought there was very little. And I wonder if rather than a $5 million single prize, a substantial number of SBIR or STTRs targeted out of this program might get you to the same end and spread this out a little more broadly so that you don't have sort of winners and losers.
Yeah, that's interesting. So to your first question, as a sensory neurophysiologist, I'm a huge fan of cochlear and retinal implants and, all manner of, transcoding gizmos. We're trying to be catalytic to all those things. The reality is that we're in the common fund lane, which we try not to operate in a particular IC's direct lane. So we have a National Eye Institute, we have a National Institute on deafness and communication. So those things are pretty well covered by their respective ICs. Now, obviously, like without my Common Fund hat on, I hope that the things that we do find their way into all kinds of other places.
Yeah, that wasn't actually the intent of my comment, but I think the challenge here is that there is a very large experiential database with these sorts of issues that I don't see a connection between those other ICs that are in that space already, just for information exchange and so forth. It's not clear to me that what we've learned about temporal versus rate coding, for example, in information transfer in the nervous system is going to make its way over to the same questions in the vagus.
Yeah, no, that's a really good point. And in terms of the X Prize and SBIRs, that's something we talk about and think about a lot. A lot of the mechanisms that we've used in SPARC don't discriminate between small businesses and academic institutions, and we've funded a bunch of small companies or academics creating small companies. So we have done that. I'm not sure that I don't believe that the OD has an SBIR sort of funding stream, so that I don't know if there's ever been a Common Fund program that operated mainly through SBIRs or an initiative, but that's something we could talk about.
Okay, I'm going to suggest that we go ahead. We're falling behind. Go ahead and take a vote, but that, Gene, you take into account these comments that have been made on challenges versus other mechanisms. SBIRs are used in ORAP. We can use them.
All right, so I'll take over.
Yes, please.
From what I heard, I'm going to ask if I may have a motion for concept clearance of the stimulating peripheral activity to relieve conditions, as in SPARC stage 2. You know the drill. I need somebody to enter into second. I didn't actually see somebody so moved. Okay, I have two. Okay, so now I see so moved and second. Thank you. Any further discussion on the motion? Not seeing anything, I'll turn it over to Christina.
Thanks, Dr. Greeter. All right, so the poll will be launching on the right, and please remember to hit submit to count your vote. The poll is starting now. You have about 15 seconds remaining. The poll will be closing now. I will now share the results with the meeting. The motion as stated has passed. Thank you.